Hi guys, for this webcast we're going to be talking about the eye and the path that light takes through the eye, and then the beginning of signal transduction and how that light is interpreted. So I've drawn this wavelength here, and we're calling this light here as it approaches the eye. And the first thing the light's going to hit is the cornea. That's the outermost segment here that I've drawn. And this is going to focus the light. This is going to begin the focusing process of the light. As the light passes through that, it's also going to pass through aqueous humor. Aqueous humor, which is a fluid that fills the anterior segment of the eye. The anterior segment. And this is very fluid-like fluid. It's not gelatinous. And the anterior segment I mentioned is going to be between the lens and the cornea. The lens and the cornea. So let me label the lens here as well. And then once that light has come all the way through the aqueous humor, it's going to hit the lens, and the lens is going to focus that light even more. And the lens is going to be the final focusing point for the eye. And it's going to be the boundary for the posterior chamber. So the segment, excuse me. Here's the anterior segment, right here, and then here's the posterior segment. And this posterior segment is then filled with a different kind of fluid called vitreous humor. And this vitreous humor is much more gelatinous than the aqueous humor. And it serves three main purposes. And that's to help transmit light, support the posterior surface of the lens, and to help maintain intraocular pressure. So pressure within the eye. Vitreous humor. And you have the same vitreous humor that you had when you were born. Whereas the aqueous humor has a high turnover rate, and you're continuously reabsorbing and then making new aqueous humor. So as that light passes and gets focused to the lens and passes through the vitreous humor of the posterior segment of the eye, it's going to hit the retina, which I've colored red back here. Retina. And this is where we're going to begin the signal transduction process. So I'm going to scroll down here. I've drawn a retina here, and it's just in the other direction. So instead of, the cornea would be here, and then the lens, and then so we're just in a different orientation here. And when light hits the retina, it's going to activate photoreceptors. And there's a couple different kinds of photoreceptors. We have rods and we have cones. And cones are what are mainly active at night. They can sense small amounts of lights, and they work in shades of gray. So we're going to have these cones here. I'm just going to begin drawing them here. I'll just draw two cones. And what I'm showing you here is that they're facing towards the retina, and so it's almost as if they're facing backwards. If you're thinking of of everything that if everything was meant to be facing forward, these neurons are facing backwards compared to everything else. And these are the rods. Now we have three different types of cones that are involved in vision processing. There's actually one more kind that has to do with circadian rhythms. But you're going to have a red, a green, and a blue cone. And there are many of these, but these are the three different kinds. So you have the red one here. Let's make it a little bit lighter red. A blue one. And a green one. And these photoreceptors are you can think of them as neurons, but they almost look like, as your book describes them, inverted columnar epithelial cells that are just facing the wrong way, or facing the opposite way of what you think they would be facing. So after these the light activates these photoreceptors, that signal is then going to be passed on to the next cell, which is bipolar cells. Bipolar cells. I'm not going to get fancy with the way I'm drawing these, so I'm just going to draw them all in one color. Here is purple. And the synapse here, let's see, let's finish off all these neurons. So we can draw some synapses here. There we go. Bipolar cells. Right here, I'm just going to draw these as sort of a continuation of a rod-looking shape. This is the next type of cell in this line of signal transduction here. And from there, the information is going to be passed on to ganglion cells. Ganglion cells. And we'll finish those off in this green color here. And these ganglion cells are all going to converge at, that's too big, the optic disc on your retina. And that is where, as all these electrons from the ganglion cells leave the optic disc, it becomes the optic nerve. And then that's where the information then starts to be passed along to your brain to and how your mind interprets it. So let's label these really quick here. This section we have ganglion cells. Cells. And right here we have bipolar cells. And then here we have photoreceptors. Photoreceptors. We have the retina here. And remember, the light is entering the eye from this side. So we have light entering the eye from here. And it's going to pass through, hit the retina, and activate these photoreceptors. And the signal then is going to be passed back the other direction. So signal is going this way, whereas light is going in the other direction. Cool. That's going to wrap up our webcast here. We just talked briefly about the anatomy of the eye and what the light passes through to hit the retina, and then briefly went into the signal transduction after it hits the retina. Thanks, guys.